thank you so much for inviting me. May I just check that you can hear me okay?
Yes, we hear you very well.
Okay, super, fantastic, thanks. So thank you so much for the invitation to speak. It's a real privilege to be on this platform amongst the other speakers. Anthony, thank you for a lovely talk, and I'm looking forward to Piers this afternoon as well. I'm sorry that we couldn't be there. Tunis has always been a place that I've always wanted to visit, and it just got a little bit busy. So hopefully, fingers crossed, next time I can address you in person. Right, so I've prepared about a half-an-hour talk, and happy to take questions afterwards, but I've split it into two sort of angles, one of which is to give you a little bit of an idea, a taste of our area of work that my team is embarked on at the CSIR, and then also to talk a little bit about how my team have and continue to think about how to do CRISPR in a resource limited setting. So that'll be the two angles of the talk that I'm gonna focus on. For those of you who don't know, the Council for Scientific and Industrial Research is South Africa's government research institute. And we have a mandate to it doesn't work quite like a traditional academic institution, but our mandate is towards developing a capable state, but also to find angles of R&D. So we have an unusual focus in that vein, but quite interesting in terms of impact. And so within that, at the CSIR, our team's objective is to really ensure that African genetic diversity is incorporated into global research drug pipeline development. It sounds like quite a lofty goal. Fortunately, I have some scientists who work in the team who are remarkably clever and brilliant at everything that they do. So they are moving us in that direction. So forgive me, Mohammed, I'm not too sure of the broader background expertise of the audience. So I'm going to start with some relatively simple genetics. And if there are any geneticists in the room, please, right from the get-go, I'm using this as the simplest possible explanation to start off the talk. So please don't think it's absolutely accurate. We have boxes of sweets in South Africa called Smarties, and I think they would probably be the equivalent of M&Ms in Europe, and I'm not too sure what Tunisia has. But if we imagine a box of multicolored sweets and we sort of put those on a map of the world, sorry, I beg your pardon. I just want to make sure that this, here we go. Africa is the origin of all human genetics, really. And so it has the whole box of Smarties, all the colors. And then about 100,000 years ago, a small group created the out of Africa migration and then subsequently populated the rest of the world. Now, again, it's really the simplest version of what could possibly, of an incredibly complex migration. But if you think about it, anyone who went left of the Caucasus Mountains, the blue Smarties or blue sweets, became Caucasians. Now, obviously, we now understand and we appreciate the fact that those of European ancestry or European descent primarily dominate the populations in Europe and in North America. And the consequence of that is that the drugs that are developed on those populations do not suit the majority of the rest of the world. So let's just take a look at what the going from a Smarties indication of what's happening to more scientific validation of that idea. So if we want to try and understand a greater idea towards pharmacovigilance in countries like South Africa, pharmacovigilance being the ability to be able to have a heads up or an understanding of adverse drug reactions in the population, we need to understand three key things. The first is that it's well established now that African genetics is by far the most diverse across the world, as much as 25% more genetic variations than the rest of the world combined, and that's pretty much dogma now. You combine that with unique and disproportionate disease burden. So by that, if you take World Mapper, I find this really helpful in understanding it. But if you look at our gross national income compared to the rest of the world, and you layer that with, for example, HIV, you can start understanding how difficult our resources are to tackle huge and relatively unique or prevalent rather disease issues. And then of course layering that with the knowledge that clinical trials are really not represented on the African continent. And so the consequence of that is that we understand that as many as 1 in 12 people are admitted to hospital for adverse drug reactions in South Africa, and that's admitted not because of the disease, but because of the drugs that they are taking. So forgive me, sorry, Mohammed, I can hear noise in the background. Does someone want to ask a question?
No, sorry, go ahead.
Okay, all right, super. So with that in mind, we need to start thinking a little bit more about how to address this kind of issue. And my team thinks about this a lot, framing the healthcare landscape for Africa. So how can we incorporate this African genetic diversity and bring that into, we use the phrase precision medicine a lot, and it's a lofty goal and I think it's amazing, but I think from a resource limited point of view, my team will often talk about stratified medicine. So stratifying based on different demographic groupings of drug patient pairing and how to improve clinical outcome. So what could we do to contribute to that particular issue? And we really see this as the solution is in developing cellular models that are physiologically relevant and genetically representative of the population. So we are moving towards developing an African hepatic modeling platform. For those people in the audience who've worked with a lot of cellular models, there's a lot of work obviously going on and immortalized cancer type cell lines, but we're really focusing, and the talk is going to primarily talk about our genome engineering work that we've been doing on induced pluripotent stem cells. But they're also, of course, the gold standard primary human hepatocytes that can answer different research questions or answer different issues of adverse drug reactions. And so the goal with this is then to go, well, can we provide our health regulators, can we provide the people who with evidence so that they can action pharmacogenetic interventions, for example? Can we work with the pharmaceutical industry to provide them with tools at multiple stages along the drug development pipeline, such that they might be, that when the drugs finally come to regions in Africa, the drugs have already taken into account, that testing has already taken into account African diversity. So that's the concepts that we focus on. I just want to mention briefly something that I was incredibly proud of. My team work led by Dr. Harrell and Dr. Naiju in localizing primary human hepatocytes extraction on the continent. So these are, for those of you who are not familiar with this particular technique, primary human hepatocytes are the gold standard for many aspects of liver modeling. But the difficulty that we have with this is that there are very few, if any, that are of African origin. And so what my team was able to do just over a year ago, there's Dr. Harrell with Ms. Shabalala, was able to localize this technology and demonstrate that these are functional. And in addition, this really helps us elevate the work that we're doing.
No, it's okay.
Sorry, I can just hear that someone isn't on mute yet, so I'm just gonna if I could just check. Super, thanks. Where was I? Right, okay, so primary human hepatocytes, and so that's something that's that we get really excited about, and how this could integrate into our stem cell work. So there is that. So if we then go back to then talking about the iPSCs, and this is largely where our area is focused on. For those of you who are not familiar with our stem cells work, I'm going to go back to a very old slide, and this was done by Waddington in the 1950s, and he created this imagery of what you could do with different stem cells. And so if you imagine yourself as a ball at the top of a series of mountains and valleys, and gravity is your only force, as you move down these different valleys, your fate to a degree is determined. So if you land up here about halfway down, you could still land here or here, but you essentially can't land up at a different value. And that's kind of how stem cells work. So if you have pluripotent cells here, you can turn them into just about anything in the lab, and we do. We played around with a little bit of neuronal differentiation. We've also generated, and I'll show you this a little bit more, apologize for the sound, sped up macrophages from our stem cell lines. And of course, one of my absolute favorites are the beating cardiomyocytes. And it's not a focus area of ours, but it does give you an indication of what you can do. So how are these generated? Well, take a look at a friend or a colleague next to you with a freckle. And in essence, that's how induced pluripotent stem cells are generated. You could take a dermal skin biopsy about the size of a freckle. Those dermal skin cells can be expanded in a dish. And that's what Shinya Yamanaka won the Nobel Prize for in 2012 from his demonstration in 2006 and 2007 that you could actually reprogram a skin cell, a terminally differentiated cell, back up Waddington's landscape all the way to the top of this pluripotency. So what my team has been working on is trying to decide how could we take these skin cells of African origin and differentiate them into liver-like cells. We started off playing around with some relatively simple methods of creating hepatocyte-like cells in a dish in 2D. But like many individuals, it's increasingly obvious that three-dimensional models may well create increased accuracy or more increased maturity or functionality. And so we have generated 3D models of various kinds, and these are quite fun actually. When the team looks at them, we see very different cell types created in these stem cell derived models. But whether you're looking at 2D or 3D, and it's all about the research question here, one of the things that you need to be able to demonstrate is that they're functional. And so that's what my team was able to do, having set up an LC-MS method of a number of different really important cytochrome, I beg your pardon, metabolizing drugs. And one of those, midazolam, which is an indicator of an active sub-3A4 pathway, was shown last year to be functional. And this was really important for us. I mean, the team's obviously coming up with some really interesting ideas about how to maximize the functionality of the other genes, but this particular gene, sub-3A4, is responsible for the metabolism of about 50% of the drugs in the market. So already they've made significant advances in creating this as a potential service for screening altered metabolism of different drugs. But the thing is that we're in a resource limited setting, and we're not going to be able to generate iPSCs from every single individual on the African continent to take that kind of diversity into account. There are a number of published papers that have indicated that there are key genetic variants in the African population in these critical metabolizing genes that would allow us to highlight these high impact ones and start off with those as we know that these ones have been shown in the literature to alter metabolism, so we can identify individuals with those. So you can do that. But we also need to understand that there might be others that are coming through that haven't necessarily been picked up. And so we were lucky enough to be given a small grant by the Gates Foundation to combine information from hospital dispensary data, but also most importantly, adverse drug reaction data specific to the country. And with that, and taking a look at those the drugs that are coming up as the high impact drugs, we've been able to extrapolate to a small degree. A lot of this is very early data. So I want to make that disclaimer, but actually using information like that, sort of look from a different angle of saying, can we identify these high risk, if one could use that phrase, genetic variants that might be prevalent, causing problems on the drugs that are most prescribed or most frequently creating adverse drug reactions. So we have a unique way of being able to analyze that. But again, it goes back to this issue of, are we going to find individuals who have these drugs? And what you can do a lot more easily, and Anthony explained the ease of use of CRISPR is you could actually use CRISPR genome engineering to create isogenic lines that represents these troublesome genetic variants that are creating a problem in our population relevant to the drugs that are the high impact adverse drug reaction drugs. And so that's where we are using, we are exploiting CRISPR as our sort of flagship project. So there's a lot of work that has to go into that, and again, and Steph, I really appreciate it because you've explained CRISPR, so I don't have to go into the background of that. But what I wanted to do is, wrapping up, this is where we are. This is the work that we're focusing on. Is talk a little bit about what we've done so far in the lab in terms of addressing aspects. Just before I get to the resource limited CRISPR techniques that we've done. So just to give you an idea of how far we are from this, the Human Pluripotent Stem Cell Registry, of those with ethnicity that have been reported, only 3% have been reported to be of African origin. So if you then take a look at that from a map point of view, you really start getting an understanding of how important these kind of African cellular models would be in contributing to these as research tools. And if you want to look at the actual science and just see this categorically from a genetics point of view, we had some of the lines that we've generated and also our primary human hepatocytes genotyped. If we look at the gene CYP2B6, which is well established for breaking down the drug, the antiretroviral faravirins. If we look at one of the alleles, which is star 6, which creates a gene that a version of this gene that makes it very difficult to break down this drug. You can see that mutation is relatively rare, 15 to 20% in the European population, but upwards of 40% in the African population. This goes back to thinking about our resource limitations, but focusing on diseases that are relevant to the region, because if you look at this particular this gene, it's not even that the difference in the allele frequency is maybe double, so it's not a massive difference, 20 to 40%, for example. But the point is that efavirenz, obviously from an HIV burden point of view, this particular allele is creating a very significant issue for us from an adverse drug reaction point of view. CYP2D6 is responsible for breaking down, converting rather, the drug tamoxifen used as a chemotherapeutic and converting it to its active compound, endoxifen. Now, in upwards of 30% of the African population, non-existent in the European population, but upwards of 30% of the African population create a version of this gene that means they can't do the conversion. This isn't even really coming up as an adverse drug reaction. This is just ineffective. Imagine an opportunity where we could prevent that from happening and choose treatment option B instead of treatment option A for our patients. And one final example that I think is really important to remember, because we talk a lot about infectious disease in Africa or cancer, for example, but tacrolimus is a drug that is essential for post-organ transplant, preventing immune rejection post-organ transplant. This allele, which rapidly leads to a version of the gene that rapidly metabolizes tacrolimus, is a minor allele in the European population, and it is the predominant form in our population. So thinking about ways in which we need to address this from a local point of view, hopefully you can start saying that these, from an editing point of view, are certainly priorities that we are thinking about. Right. So that's given you an indication of, I think, where we're coming from and how we think about the work that we do. But I wanted to highlight certain angles that where my team think about how to make the CRISPR engineering work that they do less expensive. So it's the time and money problem, right? So what my team try to do is I'm going to talk about 3 examples. 1, using clever design tricks. 2, using local reagents whenever possible. And 3, and again, Anthony, you spoke about this, spending more time optimizing your design strategy right at the beginning is really important, so that you can minimize the time for the derivation of the clones themselves. So we're going to talk about those 3 aspects. Using clever design tricks, right? So one of the things that the team has introduced in the lab is something really simple, and that's just using multiple excision to establish knockout lines. And so one of the reasons why this is effective is really largely about reducing the number of clones that you might have to screen. So the concept here is, rather than using a single guide site, you use 2 guide sites, and then you get deletion between those double strand breaks. And you can visualize that on a gel. So imagine if you were just having to run this gel, you'd run the PCR product from these different clones on a gel, but you'd be able to immediately see which ones have got a deleted size just by virtue of an agarose gel, and then reducing costs to then only go and sequence the ones that have that deletion. So that's a strategy that they use. So for example, in our lab, multiple guides are designed. You can see various primers that we use to amplify those regions. And so when we're looking at, for example, a global T7 of a gene that we were targeting, and looking at these different guides, we were getting really ineffective indel efficiency. But when we combine them, you can see here, so guides one is okay, guide 2 doesn't really seem to work at all, but when you start combining them, that indel frequency really sort of leapt up another level, and combining multiple ones also subsequently led to increased indels as a result of that. But what is handy with this is that the T7 assay itself is quite an expensive reagent. So my team's going to shout at me after this for getting this wrong, but two EPIs of T7 that you order could cover the cost of an antibody, for example. So it's not an inexpensive reagent. But if you are creating microdeletions as a result of having 2 guides, you could actually just run these on a gel, and you'd be able to pick up those size differences without having to use the T7 at all. So it's a handy little trick that my team used to be able to identify how to target a gene, and specifically with regards to knockouts. Subsequently, then, of course, again, we've spoken about this. T7 is expensive, so it makes it a lot easier to identify those particular clones. So, for example, clone 51 here, the team was able to identify had a deletion, and when they ran that against the isogenic control, they were able to identify a biallelic knockout. And this is just to show the premature stop codon, how quickly that comes up in a 375 amino acid protein, so at position 48, and then on the other allele position 18. And this is something I want to reinforce here, because it came up in the previous talk about the length of time that it takes to take these to create these clones, because it does often take a lot of time. And what else have we done to the cells as we go through? So the team think a lot about making sure that that we have good controls as we go through creating wild types that are still functional as per what they would have originally done. So the other aspect that second aspect I want to talk about is using local reagents. And I don't know for the international, the sort of the European audience, if this is something that you guys use. Be quite interested to hear if you do. And again, I keep mentioning that the T7 endonuclease can be quite pricey, especially when you start talking about transport down to the tip of Africa. So it becomes a costly reagent for us. And it's relatively easy to replace that with crude extract of cell 1 endonuclease from, of all things, celery. So, for example, this is what this looks like. You can actually incorporate this into assessing both the global population and from a clonal point of view. So, for example, you're looking for indels, you want to assess the effectiveness of your guides. So you'd run it through the normal process, but instead of using the relatively expensive T7, you can actually replace that with the crude cell 1 extract. This is an example of what my team is able to show. So for example, in our quality control gene that we always target to make sure that the whole process of transfection and everything else that we're doing is effective. Using cell 1, you're able to pick up, it's within range of what the T7 would be able to do from a global point of view. So when they targeted another gene that we were interested in, and then expanded those particular clones, you can see here that the cell 1 was able to identify each one of those clones rather effectively. And just as an example, when we look at the versions of wild type I beg your pardon, versions of the wild type that hadn't got a yielded an indel versus those that did, you can see that this came up really nicely with the sequencing. So again, local reagents, celery of all things, that's something that we find that's quite effective. Right. So I just want to wrap up with an angle, and it's a project that we're working on with NICD in Johannesburg. We think a lot about how to create cellular models of African relevance. One of the areas that the NICD is particularly interested in is talking about diseases linked with immune dysregulation. And this obviously has particular relevance post the pandemic. So how could you do this? How could, given the tools that we've been talking about, how could we potentially help to try and understand unique contributions of rare genetic variants within the African population? So there's a fairly well-established technique by colleagues of ours, Dr. Sally Cowley at the University of Oxford, who was able to show a relatively efficient way of differentiating iPSCs to macrophages. It was a technique that we set up in the lab some time ago. It's a really sweet protocol. If you enjoy even phase contrast imagery of morphological images of cells, these are the monocytes that are generated from the factories. And within a week of that, you can differentiate these into mature macrophages in the dish. So it's a really useful technique. When they speed up, you can see the interactions between them. So you can actually see them sort of mopping up any of the residual debris in the plates. So they're really, really sweet to work with. If you take them out the incubator for too long, they slow down, so you have to get them back in. But using this method here, and then again, just to reinforce how we were thinking about using genome engineering in this vein, we wanted to make sure that our procedure was accurate and that these cells were functionally could functionally validate a phenotype of interest. So we took our monocytes, matured them to macrophage, and then infected them with HIV pseudovirus. And so this is what those would look like here. So these are unedited macrophages. And when you infect them with HIV, there's this really classic largely in vitro phenotype that occurs of syncytia. So you can see the multiple nuclei over here. In contrast, when we gene edited, knocked out the CCR5 gene, which is an essential co-receptor for HIV infection in immune cells, this is what they look like uninfected. So pretty much the same array in there. But when we infected them with the HIV pseudovirus, you could see that they were completely refractive to HIV infection. This has been something that's been published numerous times, but again, it was helpful for us in demonstrating that using CRISPR engineering to validate a particular iPSC-derived model should give us some confidence in being able to pursue this. So how does this then link back to this point 3 of how to study a novel genetic variant? I want to reinforce this. This is from the same paper that I mentioned right at the beginning. Genetic variants that are thought to be rare in traditionally narrow populations are oftentimes found to be highly prevalent in African populations. And again, as a result of the migration of humans over time. So what could be really helpful here is to be able to demonstrate that highlighting one of these that you could actually demonstrate an African mutation functionality. Mohammed, I can see you there. I assume that means 1 minute. Yes.
Let's say 5 minutes.
Okay. Well, I've only got 3 more slides, so we're just about there. So when we were our colleagues at NICD provided us with some information, and they'd identified patients who had a novel mutation in an immune gene. And these individuals appeared to, you can see here, they are the red dots. They appeared to have a dampened immune response when challenged by IL-6 and sorry, not challenged by, in terms of their ability to produce IL-6 and IL-8. And they wanted to understand what could possibly, if we had any of the models that would be able to show the functionality of that particular variant. So this goes back to this last little trick. And again, I cannot reinforce this enough, where my team thinks about how they can shorten the time by using multi-cited published methods that are reliable and optimizing that at the beginning. So HDR is resource heavy. It takes a lot of time to screen the clones. It takes a lot of media. It's expensive, goes through a lot of passages. So you start wondering about the stem cell's integrity. And Cas9 can be expensive, for example, if you're using an RMP method. So what did they do instead? They've used the PR or Pure system. And essentially what happens with this particular method is that you are transfecting your cells with the full Cas9 machinery. So you've got your Cas9, you've got your guide, and in this particular case, your HDR template that can go and do the repair. But what this system does is it asks you to add yet another piece of cargo, and that piece of cargo comes in the form of a plasmid. Now, from a really clever design point of view, that particular plasmid has got a puromycin gene that has been broken or interrupted rather. And the piece of DNA by which it's been interrupted is the target site of your guide RNA. So what happens is that you load all of this into your induced pluripotent stem cell line, for example, and you obviously want your machinery to go and target the endogenous loci. But what happens here is that your guide RNA will also target this plasmid. That'll lead to the puromycin gene resealing, for want of a better word, so having an intact puromycin resistance gene. So such that with the addition of puromycin, you can select for cells that are undergoing HDR, so those dividing cells, that are at a point in the cell cycle where all the HDR machinery is present. So the consequence of that means that you've massively enhanced your ability to select for clones that may then have the HDR machinery to go and create the specific repair that you want at the endogenous loci. So just as we wrap up, this is what it would look like. So if we look at HDR efficiency in stem cells, and we're talking about human-induced pluripotent stem cells, what you're going to be doing is sending your postdocs out screening for, maybe if they're lucky, between 1 and 2 clones per 100, as an example here. And you don't want your postdocs to do that. You want them to be thinking about more experiments, right? You want to reduce the amount of time. And so certainly in our hands, with Dr. Komtu and Dr. Gula, what they've been able to see is almost 2/3 of the cells that clones that they look towards are isolating have got an HDR repair. And that creates a significant amount of savings, both in terms of time and in terms of money from reagents point of view. So with that sort of rapid indication of some of the work that we've been doing and how we try and think about being resourceful, I just want to wrap up. George Box said no model is correct, but some are useful. It really is all about your research question. There's no one particular solution for developing this. You really need to think about multiple angles. And obviously, we just want to say that we'd really love to collaborate on African iPSC partnerships so that we can maximize my team's expertise in the genome engineering space. Just a little note that there is a South African conference on advanced cell models taking place in October. So I'd really encourage all of you to take a look at that, and we'd love to host you here. This is my team. Dr. Harrell and Dr. Naidu, who are leading the charge with the bioengineered liver, and Dr. Gulab and Dr. Kontu, who are doing the genome engineering. We're funded primarily from the Department of Science and Innovation, but we also have been supported by the Gates and the NRF. And the data that I showed you of the genome engineering was primarily associated with collaborations at NICD and NWU. But we do collaborate with a lot of other people. So with that, I'll pause and hopefully there'll be some questions.
Extraordinary. I have to maybe, you hear me?
Yes, I can hear you perfectly.
Yeah, so extraordinary talk. I really loved that celery can beat Merck, for example. So it's wonderful. I have, before going with the audience, I have maybe, so when you talked about multi-excision, you know, so is it in a way in your model, liver cancer or liver cell, is it, how can I explain myself in an easy way? So is it really recommended to go multi-excision when we talk about optimization or low budget low budget model? Is it, I mean, is it really recommended to go multi-excision? This is my first question.
Okay.
Back to the story of celery, because I really love the image. People here, maybe also we are a bit afraid when we go Fantasia. We are very afraid that journal will not accept this fantasy to publish. So how do you comment this? And thank you.
That's a pleasure. So I'm happy to address the first question, and then I might just need a little checking on the second one. But from a multiple excision point of view, so look, there are pros and cons to this, right? So we need to think about off-target effects. And granted, the work that we're doing is all in vitro models. It doesn't mean that you worry less about the off-target effects. I suppose you could get away with it to a degree. But with multiple guides, you're going to have multiple off-target regions, right? So there is that as a concern. I think if you're going to go with sort of a bog standard knockout and you want to be able to do that with rapidity, there's an argument to be made, and my team are online. So when I say something incorrectly, they're going to put their hands up and correct whatever I say. But the argument again is that you can, if you think about it, is that if these guides are coming together in a relatively close area, you're probably going to have more of that machinery taking place. So there's an angle to say that's partly also why you get this increased efficiency. So it's not like 1 + 1 = 2, it's 1 + 1 = 5. And that does help. And I think we always think about, the team constantly thinks about how can we minimize, in particular with stem cells, because it takes so long to create these clones, minimize the amount of passages that you go through, right? You want to minimize that because genetic integrity is really important. I don't want these things going through 20 passages in addition. They've picked up a chromosome 12, or there's some, unbalanced translocation between chromosome 20 and chromosome 19. So from that point of view, I think there is an advantage, but again, it's about the research question.
Okay, so concerning this cell 1 and T7, I mean, people here are, but also when we go with this fantasy and with this unhabitued, how to say it, with this non-conventional stuff, do you think it will be difficult later to publish with this technique, or it's just science,
So for example, the University of Michigan uses this pretty robustly. You can go onto their website and they can go and demonstrate that. I think the purpose of doing it is to identify the clones. So if you've identified the clones, you've done it, right? What's always important is that you have to subsequently make sure that you've sequenced and that you've got the right, you understand what you got. What we're talking about here is using a tool that's easily available, that's inexpensive, and helps us get to sequencing only 10 instead of sequencing 100. That's how it's being used. So I really can't imagine there being an issue from a peer review point of view.
Me too. So okay, so any question from the audience maybe? Yes, I mean, yeah. Janine, Dr. Janine.
Thanks, Janine, for this insightful presentation. My question is, how could we harness the recent breakthroughs in single-cell multi-omics atlas, which has just got out, to set the standard for clinical gene editing? Wouldn't this kind of information bring the missing piece to the workflow we're trying to improve? Thank you.
I'm afraid I didn't quite hear that. I heard something. Did it? I'm sorry. The question wasn't. Forgive me.
Which one? Eddie. Okay, so I will. So the question is, how could we harness the recent breakthrough in single cell mutation multi-omics atlas, which has just got out, to set the standard for clinical gene editing?
Sorry, are you asking how that will change what we are doing?
Yeah.
Yes.
Currently on a global level.
In terms of precision medicine, this is the
Okay. All right. Okay. So my team members are going to jump in and talk about the single cell aspects, which is something that's really exciting for us. I think to your question about precision medicine, how that would resolve that problem. Certainly from our angle, the way that we're looking at how gene editing would work, we can still extract that from looking at a global population and then identifying out those particular clones. I think where you were going with is in terms of scale as well, and understanding from a single cell level what specific edits might make at that particular point. Was that where the question was going?
I do assume yes. Thank you.
Okay.
Maybe.
Yeah.
Another question, yes.
How is the that would be useful for understanding and giving variability?
Okay, so here maybe the question is if CRISPR-Cas can help in identifying or can be helpful in metabolic disease. No, Pastor?
Oh, yeah, absolutely.
Yeah, go ahead, Janine.
Yeah, absolutely. I think, this is what's so incredible about this tool and a well-deserved Nobel Prize for Dalton and Charpentier. It's created that kind of accessibility for scientists all over the world, right? Because obviously zinc fingers and talons were pretty difficult to do. So it's as a tool, barring I think a few rules in terms of integration of particular size, but there are backs and all kinds of other things that you can do. You can really, if you know the sequence that you want to target, you can modify it in just about any way. So for metabolomic diseases, absolutely yes. There's interest in that, and it's anything that where you understand the genetic location, I think by and large, and also, if I may, forgive me, I don't know who the person was who asked. You've also referred to really lovely aspects, and I think it's coming up in the talks. It's not just about the sequence, right? It's also from a temporal regulatory point of view. Do we need to think about targeting enhancers that are not in genes? Do we need to talk about CTCF sites that control chromatin loops, for example? Do we need to talk about CRISPR activation or CRISPR-I? I think those talks are coming, so I'm not the expert, so I won't talk to that. But epigenetic modifications are extraordinary. If you think about John Rind's paper from a few years ago, where you could load DCAS9 with a long non-coding RNA and target that to a specific site, the experiments are endless. So definitely metabolic disease, yes.
Great. So how easy? Come, I mean, it will be easy. ولا تقرأ. Because here in Pasteur Institute of Tennis, there is a lot of groups, and they are really eager to know about their own context. So that's why we have this kind of.
So you have explained the cytochrome variability between African and European population. and I didn't understand well how CRISPR technique could be useful in identifying genetic variability in metabolizing enzymes. Thank you.
Okay. I mean, I think I was trying to get a lot of information across. So I do apologize if that wasn't clear. So there are pros and cons to, like any model, induced pluripotent stem cells. One of the things that has happened, and thank you for the opportunity to expand on this because I think it's really important. When I think it was Rudy Janisch as well, developed a stem cell derived model of Parkinson's disease, for example, and he differentiated the stem cells into dopaminergic neurons. He took individuals with a LRRK2 mutation. So it's well established that creates is responsible for Parkinson's disease in those patients. And then what they did was then they took iPSCs from individuals who didn't have the mutation, they converted them into neurons in a dish. But when they did a transcriptomic comparison, they really struggled to pull out any differences. And one of the reasons this comes up a lot with iPSCs is that it's just a model. These are not neurons in a dish. These are neurons from this patient. It's a model. So they don't always have the functionality that we want to look at. And then if you layer that with extraordinary genetic diversity between individuals, like he did, I think he had three patients and then 3 controls. Because of that diversity, can often dilute the impact or mask the key pathways where the differences were. And so what he subsequently then did was he genetically engineered I think he was the LRRK2 patient cells, and he corrected that mutation. Then he put those through the differentiation procedure and he got beautiful transcriptomic profile differences. And if I'm not mistaken, actually identified a novel drug target as a result of that. So to your question, in the same way, I'm not going to create iPSCs from the whole population of Africa. What I can do is use CRISPR engineering to take individuals who have got some of these genetic variants, and I say repair, that's maybe not the right word, and then engineer them to have maybe the equivalent of the European alleles, and we can run those through as isogenic comparisons. I hope that answers the question.
I do assume yes. Any other question, maybe? Yes.
Hi, Janine. I think that will be a big chance to lose control multiple decision. What do you think? We will lose the control on editing and the results.
So again, the question, again the question of multi-excision stuff. So the question is, we are a bit old. The question is, we are a bit.
We have a big chance to lose control.
Yeah. So the final tap, is it? I mean, is it really adequate as an approach to the multi-excision? This is the question.
In terms of is it accepted from a public peer-reviewed
point
of view?
I mean,
is that no?
Is there any change that we are not really? We are not getting the final tap we want with multi-excision.
Okay. Okay, so the multi-excision it's primarily restricted to knockout, right? So what you want to do with that, and again, I want to be careful about, we have very heated discussions in my lab about what is a knockout. And again, linked to something that Anthony was talking about, you know, if you've got a cell type that's essential, it might try and find a way of using a second transcription start site or translational start site. We think a lot about that. I didn't even mention that, but that's obviously something that we think about. Is how much of that protein do you have left? And so with the multi-excision, the chances of you creating a premature stop codon, because you're removing such a huge chunk, right, is that the chances of your protein subsequently having any kind of functionality are reduced. Really what you should be doing, and I'm saying this as a result of one of my team members in particular, is there should be no one of the views in my team is that there should be no RNA coming off, whatever, never mind just a truncated protein. That's something that comes up. And again, to the earlier talk about, you know, the knockout has happened, but the cell has found a way to use a minimal level. So with the deletions, you've maybe got a little bit more chance of making sure that that's not possible.
So optimization is the key. Definitely.
Yeah.
Maybe last question. Yes. 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 Bye.
Using the cassette of said that using selection, it could be.
Oh, it's a long question. Please come and do it yourself, because it's a long question and it's more technical than I thought. So, so please. And Pia, we will be just like 10 minutes late.
Can you hear me?
Yes.
Okay.
Yes, I can hear you perfectly.
Okay. Thanks, Dr. Janine, for this nice talk. My question is about the homology-directed repair and the selection of the cells who efficiently underwent the homology-directed repair. You have spoken about a pure cassette that we can use and the puromycin selection. And this could be a low-budget way to select the cells who had underwent homology-directed repair. But so far as I know, this cassette allows only to select the cells that had an efficient transfection. It means that this is the first level to see which are the cells that had been transfected and no, but it cannot be in somehow a really efficient, low budget method to say, okay, we have these cells that are that underwent efficient homology direct repair or not. So the only way remaining to confirm this issue is to do sequencing to say that we have homology direct repair or not. So I didn't understand how this method, I mean, puromycin selection, how it could be a low budget method to select the cells that really underwent homology direct repair.
Grazie.
Thank you.
Perhaps I didn't explain it correctly, and thank you for giving me an opportunity to do that. I think, so first of all, I think there was a question about transfection efficiency. You know, for example, you know, human pluripotent stem cells are not easy to transfect. And so that's one of the reasons, one of the many contributing reasons why editing is inefficient in those types of cells, whereas you do that with, you know, the equivalent of a HeLa line or something else, and you get much higher efficiency, right? So transfection is a key barrier to increasing this efficiency. The presence of this cassette, the split puro system, allows for you to enhance the number of cells that at that one particular time in their cell cycle, the HDR machinery is present. So that's another limitation in developing and creating HDR. So we know that it's almost impossible to do HDR, barring some other methods that have come through. Never mind base pairing, base editing, etc. But classically, it's almost impossible to do HDR in senescent cells because the machinery is not active. It's not there, so it's not going to take place. So what you want to be able to do if you need to do HDR is, ideally, you want all the cells to have, you know, have as many of these cells having that machinery, because if that machinery is present, then it means that at the endogenous loci, there's more chance of that repair taking place. So the split puro system, what it does is that it allows you to, because you're selecting by puromycin, if the cells have taken up the plasmid, which they should have if they've taken up all the Cas9 machinery, the Cas9 machinery will also target the plasmid itself, allow for the puromycin resistance gene to be expressed if there's HDR machinery in the cell. So it's not happening at the endogenous site at the locus itself. It's happening episomerly. So all you're doing is enhancing the number of cells that are undergoing HDR, that have the HDR machinery active at that particular point, because the puromycin is going to kill all those other cells that haven't resealed that resistance cassette. Is that more clear?
No, it is clear, but my question was the information you gave means that this way is the method to select the cells that may undergo homology direct repair, but to confirm whether the cell will effectively go with homology direct repair or not, it depends lately to the molecular screening and the molecular confirmation that your cells really underwent homology direct repair. So it could be a preliminary screening to select the cells who are potentially able to undergo homology direct repair, but not to confirm that really your cells are undergoing homology direct repair.
Yeah, no, sure, absolutely. So it's another method to get you to screen less clones.
Okay.
So as opposed to Doudna's eLife paper, where you go through a method like this and you might get 1 or 2 out of 100, absolutely. Whereas with this, my postdocs were getting 10 out of 15. It's reduced the number that they have to screen. So it's just about saving time and money. You're absolutely right. You still need to go and sequence and make sure that they're correct. Yes.
Thank
Apologies.
you so
Sorry,
much, Janine. Thank you.
Yeah.
Thank you.
So Janine, again, thank you very much. It was a fantastic discussion, and I'm sure that people here will continue to email you and ask for other questions, etc., etc. So thank you very much.